Do you? I was gonna ask you about it a little later into to hear that. It's been working really hard to try to make it more streamlined and easier to figure out like what needs to be done this week versus like the whole unit. And that, and like, I don't know if you agree, but like the weekly assignments, like stuff is due weekly as opposed to right before the unit. You know, people put it all off till like the day before the exam. So now at least we have something due every week to keep people kind of progressively working on it instead of just all right before the exam. So we'll see. I'll probably ask you a little bit further into the semester and get your opinions on that. You're not the only person. Probably have a collection of people, a mini handful. Okay.
I mean, do you want to get like after
Yeah, always open to that. You can come by, like either stop me after lab or just come by during office hours or email me and we'll make time for it.
I think I have a better understanding.
Yeah, now you kind of know what to expect and you kind of know what to do differently in this semester and stuff. So that'll help. Make sure you're getting signed into the Kahoot. We're gonna get started here in a couple minutes. Everybody kind
of your Eukaryotes. Yep, I can't speak.
You're okay.
Anyway, chapter 5, right?
Chapter 5 for the eukaryote.
Yeah. I figured, but
For the quiz, were you asking, or for the lecture?
oh yeah,
The quiz will be chapter 4, bacteria. So remember quizzes?
yeah, quizzes are
The previous chapter, exactly. The previous chapter for quizzes.
I just messed up writing the title.
Oh, you're fine. For
So,
your notes.
yeah.
While we're waiting on the last few people to kind of trickle in and join us, I want to let you guys know I did post the weekly review that we did on Friday. That video is posted now in the discussion area. So if you guys are looking at those, as well as the AI generated summary of that review that we had on Friday. So that should be available for those of you guys who are following on the weekly reviews. Remember, if you haven't been joining or looking at the weekly reviews so far, I know we only had 2, but if you haven't been looking at those typically we just run through the major topics that we cover briefly summarizing those. And then we go over the most missed topics of the exam. So that means people who like 50% of the class or less have passed those questions on the exam relating to that topic. So we went over chapter 4 this last Friday. On previous Friday was chapters 1, 3, and 2. And so this Friday, 10 a.m. again, Zoom. And like I said, I always record it and post it afterwards if you can't make it, as well as the summary. That's going to be over this week's chapters, 5, the eukaryotes, and 7, which is going to be our viruses. So keep forgetting that this is a different screen now. We'll let everybody get signed in on this. Make sure I'm not missing any chats. Actually, open this and drag it over here in case we get any. So that you guys are prepared for the quiz. I know that even though this is participation based, you guys are always welcome to use your notes to answer these. Remember, it's open notes. So this will be chapter 4. We're going to be having our pre-lecture quiz over our previous chapter, chapter 4, which is bacteria and archaea. Engaging with some of this material, and then we will jump into chapter 5 lecture, which will be over eukaryotes today. I know we have a couple people out for the flu. Like I warned you guys last time, flu is really going around. So keep an eye out for symptoms or anybody in your family or that you work with closely that has the flu. So you can keep track of that. And you can always buy, now they don't just offer COVID over-the-counter tests for home tests. Now the ones you can buy now that have COVID and flu A and B on there to do at home. So it'll save you a doctor's trip at a copay. You can get those at CVS and Walmart and stuff, if you didn't know. All right, everybody ready? Anybody waiting to get signed in? All right, let's go ahead and get started. Right, true or false? Since bacteria don't have a nucleus, they can't have DNA. Is true or false? All right, so this one is false. Their genetics are still DNA based. They have a genome that is circular. It's a one circular piece as opposed to ours where we have multiple linear chromosomes. So one big circle for most of these guys, it's still DNA. They just don't have a membrane-bound nucleus. So yeah, they still got DNA. Our next question. The peptidoglycan layer is blank in gram-positive bacteria compared to the gram-negative bacteria. All right, so most of you guys got this one. Good job. So this is going to be the gram-positive bacteria have the thicker and substantially thicker. We're talking about 10 to even up to 80 times as thick of that peptidoglycan cell wall. So gram-positives having that thick one. Gram-negatives have a much thinner cell wall. And so what else do they have to stabilize their shape? Does anybody remember? They have an outer membrane. Exactly. Good job. I'm going to use my phone as a mouse. All right, guys, as long as you get signed in, remember, as long as you get one question in, it'll count for your attendance. Gram-negative outer membrane contains which of the following? All right, so for this one, that gram-negative outer membrane has something in it that chemically we refer to as lipopolysaccharides, right? And an LPS for short, and what the medical community has termed endotoxins. And endo because it's part of the actual gram-negative cell, and toxin because it's going to cause a lot of damage within the host, typically us humans. as far as eliciting the inflammatory response and causing fever and even shock, potentially according to whatever the reaction could be in response to it being present where it shouldn't be, and the immune system responding to that, which we'll learn more about in later chapters. These other choices that we have available on here for exotoxins, these are toxins that can be secreted by the bacteria outside of the bacteria and doesn't have anything to do with our outer membrane, with our gram negatives. And then we have the lipoteichoic toxin. So lipoteichoic and teichoic acid, these are structural molecules that help with that thick peptidoglycan layer for the gram positives. So that's not important for the gram negatives. All right, so we know what's going on with those other choices. Moving on to our next question, number 4. What is acid-fast staining? The best answer for this one. Okay, so acid-fast staining is a stain to determine if we have mycolic acid present. And we'll talk more about acid-fast staining in lab 4 in the lab whenever we get to actually do acid-fast staining. But basically, what's going to be involved is a lipid-soluble stain, and that mycolic acid is actually a wax. It's a fatty acid heavy wax, and it's very hydrophobic. And so that's going to be very water-fearing and even charged molecule-fearing in general. So we use a special lipid-soluble stain for that helps us see if that wax is there. And if we remember correctly, what type of bacteria tend to have mycolic acid or be acid-fast positive? You guys remember what they're called? Mycobacterium, right? And so those are going to include the cause of tuberculosis and leprosy. So that's going to be the answer for acid-fast staining. Question 5, true or false? Mycoplasma is an acid-fast positive organism with a thick, waxy cell wall. Now, if you paid attention to what I was just saying in the previous question, then maybe you would know the answer to this one, or maybe you're still confused. So take a minute, look it up in your notes. No worries, if you get it wrong, I'll tell you what the answer is and why. So mycoplasma is not acid-fast. It does not have a waxy cell wall. The mycobacterium have that. So remember how I was talking with you guys in the last chapter about how the way to kind of remember the difference between these guys? Mycobacterium, the bacteria that have the mycolic acid. Mycoplasma, you can kind of in your minds strike out the myco part, okay? The plasma, to me, how I remember if I'm carrying around that cup of plasma, somebody's actual literal from their blood, liquid portion of their blood, and spill it, it's not going to retain its shape. Neither do these cells. Why? Because they lack a cell wall. And so that's how we're going to, how I differentiate the difference in my mind between those two major kind of deviations from the typical bacterial cell wall. All right, question 6. What are bacilli? What is this term referring to? Okay, so the two main shapes of bacteria that I told you guys to be particularly aware of were the bacilli and the cocci. Remember the cocci, C-O-C-C, that those are very round letters. So those are going to be our round cells, our spherical shaped cells, versus the rods, our long, like kind of linearly shaped cells. Those are our bacilli. And those two L's can help you remember that those are the rod ones. So that's how we're going to try to help remember the difference between those. Most of you guys got this one. Great job. And that's referring to cell shape. Question 7, what does peritrichous mean? What's that referring to with our bacteria? Those of you who came in a little later, did you guys get signed in okay? Anybody have any problems joining in? Okay. All right, so peritrichous is referring to flagella being distributed over the entirety of that bacterial cell. There's 4 different arrangements that we can have if you have flagella. So remember, not all bacteria are motile, and therefore not all bacteria have flagella, and flagella being the only appendage they can use for motility. So that would be cell lacking flagella would be non-motile. It just wouldn't be any kind of trichus. So the 4 arrangements of flagella, and again, trichus meaning refers to hairs, right? And these flagella really quite resemble hairs. We have monotrichus having 1 flagellum. We have amphitrichus, flagella at each pole of the long cells that actually have poles. And then tuft of flagella at one end, that's going to be lophotrichus. And then peritrichus is all over. That's all of them. Okay. Moving on. Question A, bacterial capsule. So which of the following best represents what the bacterial capsule is? Hey, great job, guys. So the capsule is really kind of for 3 major purposes for our bacteria. It's that sticky, sugary coating. I kind of equate it to if you were covered in a layer of like honey or something, very sugary, sticky, allows for adherence to surfaces, allows for creating biofilms. So when a bacteria are living in like big groups and colonies with one another. And it also allows for evading phagocytosis, phago being eating, cyto being cells, when our white blood cells are trying to eat up any foreign bacterial or cells or waste or whatever they find, that's called phagocytosis. And that sticky coating of that capsule helps them evade being eaten up by our white cells. Next question. Question 9. When are endospores produced? Okay, so this is a popular choice that seems to happen from semester to semester. Let's remember that endospores are a structure that bacteria make in order to basically hunker down in harsh conditions. So most bacteria actually prefer to live in moist and warm conditions. That's a favorable condition for a lot of bacteria and a lot of microorganisms in general. So they won't need to make endospores typically in the moist, warm conditions because that's a favorable condition to them. Instead, when we make endospores, when we want to hunker down, become dormant, and wait until things get favorable, it's usually when we are lacking nutrients, or if we're exposed to something that is deadly to us, like what we see with Clostridium bacteria that are anaerobic, they can't survive with oxygen. So they will go dormant and wait until they get into an area where there's no oxygen, and then come back out and become active, multiplying, dividing cells again. Do you guys remember the term for those active multiplying cells? Vegetative, right? So those are metabolically active and reproducing. Endospores are completely inert and dormant, and that's only because they're lacking nutrients or water, or they're in some other unfavorable conditions. The best example here would be in nutrient deficient conditions. It's not a means for replication, as one cell will make one endospore. So we're not actually reproducing this way. We are maintaining our numbers, but just preparing for harsh conditions. Right, last question for the quiz. How would you describe strepto arrangements? Where there are 3 really major new terms that we learned for our arrangements. We talked about diplo, we talked about tetrad, but those words kind of have their definition pretty much in their names. So the other 3 that we had to learn were strepto, staphylo, and palisades. So palisades are only going to be with our bacilli, and they're going to be our fence post arrangements next to each other. Then staphylo and strepto. Staphylo is going to be irregular or grape-like clusters, whereas strepto is strips. I remember the difference. Strepto sounds like strips to me. They are strips or chain of those cells linked end to end to one another. So that's where most of you guys got this one. That red choice, chains, is going to be our correct answer. Let's see our podium, and then we'll get into the lecture. So good job. So for today, for our lecture questions at the end of the lecture that you guys are going to be responsible for question 1, we have Jordan. For question 2, I rolled 26. We only have 24 people in the class. So I will cover for question 2. For question 3, that'll be Jessica's responsibility. Question 4, Reagan. And question 5 will be Katie. Okay, so you guys can prepare for those questions at the end of the chapter. Righty. Good job, guys. You guys did pretty well with that. All right, let's move into our lecture. So today we're going to be getting into the eukaryotes. And so we talk about bacteria and archaea. They don't have a nucleus. They don't have any membrane-bound organelles at all. But what really are organelles? Well, we're going to get into that today. We're going to get into some examples of them and what those structures are and what they do for the cell as well. But first, when we start thinking about where life came from and where these cells came from, there is actually quite a lot of evidence that suggests that our cells that have these organelles, these membrane-bound functional units in them that have these designated functions in the cell, those organelles may have, or at least some of them, may have come from previous organisms being used by other organisms inside to create energy for them, for example. And that this relationship that we might have called symbiotic, that we have one cell inside performing this function for the bigger cell that's containing it, well, the one inside is getting protection, it's getting nutrients from that bigger cell, and it's not having to go out and find these nutrients. Whereas whenever we're talking about that little cell's providing energy product for the bigger cell that it's being contained in. And that over time, this has become what seems to be a permanent relationship. And there's actually a lot of evidence that supports this. And that concept of all that, what I just explained, is called the endosymbiotic theory. And we think this is where some of our organelles may have come from. We're going to talk about it periodically as we get introduced to some of these organelles that have that evidence, the mitochondria and the chloroplast. They're probably our biggest examples of these. We'll talk about their differences as we get to them. So we have some structures that are present in all eukaryotic cells, and we have some structures that are only found in some of our eukaryotic cells. So here on the left, all eukaryotic cells have a cell membrane or cytoplasmic membrane. We learned about this in bacteria, right? The cytoplasmic membrane contains the goo and everything inside of the cell, right? So that's the membrane that is in contact with everything, all of the cell contents in that cell. So that's the cytoplasmic membrane. Then we have the nucleus, the membrane-bound organelle that's going to contain our genetic information, the mitochondria, the endoplasmic reticulum, the Golgi apparatus, vacuoles, the cytoskeleton, and the glycocalyx. Again, we've talked about glycocalyx with our bacteria, and it turns out our cells can have one too. Now we're going to talk about the functions of those guys in the middle here in a moment. So all eukaryotic cells have to have those structures. Some eukaryotic cells may have a cell wall. So we're not really going to talk about plants. Sorry, Justin, I was going to say, we're not going to talk about too much about plants today because we're focusing on our microorganisms today. But plants have a cell wall. Most of us are aware of that. It's how they get their rigid structure and can maintain these tall trees or even support these big flowers. And that rigidity of those cells comes from their cell wall, that cellulose. It's actually where we get a lot of our fiber from our diet and bulk up, your stool so you don't have too loose and so it keeps you regular. So that's about the idea with the cell wall for plants, which we're probably more commonly familiar with. But we do also have cell walls in our fungi. And fungi can have cell walls made of cellulose or chitin, which is something, maybe a molecule you're familiar with insects. We also have locomotor appendages. These guys aren't just going to be defined by only having flagella. These guys can have flagella, but they are much more sophisticated than the bacterial flagella. But they can also have things called cilia, which are smaller hair-like appendages that help with movement of typically fluid that can help with feeding. They're like oar-like appendages, little tiny ones that can help with feeding. And you'll have cilia on the cells in your intestine that help move fluids and things like that through your intestine, through the solid matter, and also help absorb nutrients through the membranes of those guys. So this can be useful for locomotion of the cells, but also maybe locomotion of material around the cells. And then we can also have chloroplasts. Chloroplasts are not just designated only for plants. Algae are actually probably one of the most important photosynthetic organisms on the planet and contribute to about 70% of our oxygenated atmosphere pretty heavy stuff for microorganisms, right? So we're going to get into these structures next. This is a great infographic to refer back to see where these structures are in the cell, and maybe how they align with one another in their functions when you guys get to studying. It also shows a relatively scaled example of bacteria compared to a typical eukaryotic cell. We're talking about roughly like 10 times smaller on average are the bacterial cells. So much smaller because they don't have to accommodate these membrane-bound structures. All right, let's start with the flagella. These are going to be our motility appendages. Now, we talk about with bacteria, the flagella is that protein that can basically turn clockwise or counterclockwise. And that's what it would have been, reserved to for bacteria. But for eukaryotes, they have a system of microtubules that run inside of it, and it's all covered by the same cytoplasmic membrane all the way around the outside of flagella. And they can actually move these in a whip-like fashion. So we have much more directed movement with our eukaryotic cells than we typically would with our prokaryotes or eukaryotes, like our bacteria and our archaea, okay? So this is a little bit more useful for them controlling their movement when they are single-celled organisms. We do have single-celled organism eukaryotes. We often think about ourselves as we're eukaryotes, we're multicellular, therefore maybe all of our eukaryotes are multicellular, and that just simply isn't true. So we're going to get into those unicellular guys today. And we talked about the cilia. These work like oar-like motion, tiny little hair-like appendages on the outside of the cells or on the outside of these multicellular structures that can help move around the fluid or the material that's outside of the cells, as well as help with locomotion when we have single-cell organisms that can be even propelled by many of these guys. So we can find these in one group of protozoa, which are single-cell organisms. But otherwise, we can find these in multicellular organisms like us and helping with our digestive movement as well. All right, moving on to the glycocalyx. Remember, glyco means sugars, and calyx means covering. So we talked about the capsule. We actually did pretty well with our question on what the capsule is for our bacteria, and that's a type of glycocalyx. In fact, eukaryotic single-cellular organisms can have a capsule as well. So that sugary, sticky coating that would help for adhesion and things like that. But in fact, in multicellular organisms, one of the things that helps keep these cells together and give structure and relatability within an organ would be our extracellular matrix. And some of your extracellular matrix actually would be part of your glycocalyx of your cells. So things that would be sugary or things that would be made up of other molecules, things like hyaluronic acid and other molecules like that, creating that extracellular matrix is actually technically part of our glycocalyx, our outer coating. It's just how it's going to work to create definition between tissues, helping keep cells attached to one another and creating our organs as single units within our whole organism. All right, like we said, some of our organisms in eukarya will have cell walls as part of their cells. We have cell walls that cover each of our individual cells. Yes, the plants, but we're focusing here on the fungi and the algae. Again, similar function as what we see with the bacteria, provides cell shape and rigidity and things like this. And since we often see things like algae and certain molds and things like that being exposed to water, having a cell wall and giving that extra rigidity and shape, when we have differing tonicities of water and maintaining if it's super salty or if it's not salty enough compared to the inside of the cell, maintaining the shape of that cell to help with survivability in different conditions. All right, moving on to that cytoplasmic membrane. Again, that phospholipid bilayer. We have our phosphates with that negative charge on the outside and our lipids that are hydrophobic on the inside, facing them. Now, for us, eukaryotes typically have sterols of some kind in their membrane to help with cell shape and rigidity. And that's going to give us characteristic shapes for cells that make up certain organs or certain organisms. So those sterols for us and our cell membranes as humans, as animals, we have cholesterol as one of those molecules that can help with the shape and stability of our cytoplasmic membranes. Whereas fungi, they have ergosterols, for example, that these can actually serve a similar purpose for their membranes. There's actually a type of fungus that can have very hallucinogenic properties called ergot that's associated with rye, and that fungus, that ergot being related to that ergosterol that is associated with that fungal membrane. named relating to that anyways. So these are important for stabilizing our membranes. And again, just like with the bacteria, the membrane is selectively permeable. This means that water and certain tiny nonpolar molecules, they can travel through the membrane freely, but anything else that wants to cross inside or outside of the cell will probably have to be controlled by a channel, usually formed by proteins and things like that. And that's how we control what goes in and out of our cells and can maintain, keeping all of our nutrients in. We're going and gathering up new nutrients from outside of the cell. All right, moving on to our actual organelles within the cell. Our first major organelle, probably the most defining organelle of a eukaryote or a true nucleus cell. This is the nucleus. This is the control center of the cell. This is a membrane-bound structure. When I say membrane, I'm always talking about phospholipid bilayers, okay? So these are contained by a phospholipid bilayer called the nuclear envelope. And inside of that nuclear envelope, we have our DNA. And within all of that mess of DNA that's contained in there, and all of those genes that are going to code for all those proteins, and the genes that code the genes that code for expression of those proteins, and everything relating to that, all hanging out in that nucleus. And we need to be making protein so much that the structures that we need to make the protein are ribosomes. We have a little center inside of our nucleus called the nucleolus, where we are making a ton of usually ribosomal RNA. We need so many ribosomes all the time that we're always making ribosomal RNA. And we can actually get a special stain in there that stains ribosomal RNA, and that little area is called the nucleolus. It is not membrane bound. It's just a region of the DNA where we are having a lot of ribosomal RNA synthesis. And those are going to form the structures that are going to be important for making our proteins. We're going to talk about that in chapter 6 in a lot more detail. All right, moving on. It's important to remember that whenever we're making copies of our cells, so you fall down and you skin your knee, and you need to create new cells to help heal up that injury, right? And we're usually just making copies of the cells nearby. Anytime we're making direct copies of our cells nearby, or we're going from a baby to an adult and growing new cells and new copies of those cells, this is called mitosis. Now, we as humans might have 23 pairs of chromosomes, and so we have to make sure we're making enough copies of those in order to get our new cells to have all of their chromosomes appropriately and everything like that. So we go through a process called mitosis to get there. And the 4 main steps of mitosis, I remember, and I'm not gonna lie, I stole this idea from Dr. Shearer. So if this is inverted, let me turn this around. I stole this idea from Dr. Shearer because I thought it was cute. Maybe you guys will think it's cute too, or maybe you'll find it useful. But the way that I remember the 4 major steps, there are intermediate steps, but the 4 major steps of mitosis is with this kind of phrase of pee mat. So if you have a puppy, you want to pee on the mat, right? So that's how he always phrased it, and that's how I remember it. So pee, this is how I think of it's like P is for prophase. And prophase is the prep stage. So here we're just getting everything ready for dividing up into 2 new cells. So we've made copies of our DNA and we've condensed it down to a very condensed form so we're not making a mess and having DNA spread everywhere. Because once we break down our nucleus and it's not contained in our nucleus anymore, when we're dividing our cells, we can't have it everywhere and getting all mishmashed up with everything else. So we condense it down to a structure called chromatin. This is actually the best time to look at your chromosomes in your cell because it's so condensed, it's easy to see them with microscopes and staining. And so typically when we're used to seeing our chromosomes as like XX, this is actually a chromosome that would normally look like this and this, one from what we call the maternal side and one from the paternal side. And you will have one from your mother and one from your father, one from the egg and one from the sperm when they come together during creation of a new cell. And that when we go through mitosis, we're actually making a copy of that chromosome. So you could think of it as making a copy of the maternal and a copy of the paternal, and they're just linked together. And they just look like X's because we've got 2 copies of each, and we're getting ready to pull them apart. So each new cell will need 1 of each. Starting to make a little sense why we have the X's there. Our chromosomes don't normally exist like that in our cells, only during mitosis. Okay, so our linear chromosomes have made copies, and we've consolidated down into chromatin, and we started making structures needed to start splitting up our cells into identical daughter cells. This is prophase. That's the prep phase. All right, the next step is metaphase. This was number 1. This is metaphase. This is number 2. And metaphase, the way I remember this one, is we're lining up our chromosomes in the middle. So we're getting that middle alignment, arranging our chromosomes right down this nice, pretty straight line, roughly down the middle of that cell, getting ready to pull those copied pairs apart from one another, okay? So this is our metaphase, lining everything up in the middle. The next phase is the anaphase. And I think of this as apart. This is where we're actually going to be splitting those paired copies apart from one another. And so this would be step number 3, anaphase. We have early and we have late, but they're both going to be associated with pulling those pairs apart. And we're getting ready to split that down the middle and create 2 cells. So we have 1 copy of your maternal on one side and 1 copy of your maternal getting pulled to the other side, okay? And then our last step is called the telophase. The best way I can kind of relate this one would be going to be that we're getting totally done with this. And so in telophase, now we've got that full separation, and we're going to reform our nucleus around those separated copies. So that's the telophase. So we can be totally done, and we'll create our nucleus to pinch off our nucleus on each side. One for one daughter cell and one for the other daughter cell, and we'll pinch off our cytoplasmic membrane in the middle to create 2 distinct daughter cells. And now each of those daughter cells would be able to then go through this whole process again if they needed to. So that's the whole idea with mitosis, creating identical daughter cells. And when we're creating our sex cells, this is a little more complicated. We're not going to go into detail on this one, but that process is called meiosis for our sex cells. And obviously, if you have a sperm, and this guy has 23 individual chromosomes, and then the egg, if it had 23, then these could come together and make 23 pairs. But if you had 23 pairs and 23 pairs coming together, well, that's 46 plus 40. That's too many. So when we're creating our sex cells, they divide out even further beyond so that they only have 1 copy of each, not 1 of mother and 1 from father, but 1 example of chromosome 1, 1 example of chromosome 2. So that whenever the egg and the sperm come together, we have the appropriate number of each, 2 from each of them, the mother and the paternal. Okay, so like I said, we're not going to go into the details of meiosis here. Well, you guys will learn a little bit about that in physiology. But that is the idea of, especially with mitosis, really important for asexual reproductive means of some of these organisms we're going to talk about. Okay, so that's mitosis. All right, moving on. So that's going to be talking about our genetic material. Now we have another structure here, the endoplasmic reticulum, or the ER. With the endoplasmic reticulum, we are talking about structure here that is used primarily for creating protein and other molecules. So we have to manufacture a protein and other molecules for the cell going on here. In our rough endoplasmic reticulum, it's rough because it is studded with these ribosome structures that we've been talking about. And do you guys remember what ribosomes are for? Making what? Proteins, yes. So proteins are using amino acids to build our proteins. Very good. So I heard some of you guys saying proteins out there. Exactly. So these are the machines that are going to be building our proteins. We'll learn about this in chapter 6 in more detail during transcription and translation. But these are the structures that are going to be doing that. And so in the endoplasmic reticulum, we have a lot of folded up membrane that is just studded with these ribosomes, ready to make protein, ready to read our basically our working copy of the gene, our messenger RNA, and they're going to read that and crank out protein while it reads it. And so this is all going to happen on the rough endoplasmic reticulum. There's parts of the endoplasmic reticulum that is smooth. In the smooth endoplasmic reticulum, we don't have any ribosomes because we're going to be synthesizing and storing non-protein molecules. So making our lipids that we need, making our carbohydrates for storing energy and things like that, that's going to be happening in our smooth endoplasmic reticulum. And so all of the manufacturing of these molecules would be happening in the endoplasmic reticulum, right? So this is kind of showing our endoplasmic reticulum over here. You can see all these little blue dots on here. These are going to be our ribosomes represented here. This is our mRNA, which is our working copy of a gene. And they're just those blue guys coming together, clamping down and reading out those guys and spitting out protein, which is what the protein being synthesized is this little worm-looking guy coming up into the actual inside of the endoplasmic reticulum, okay? So all that's happening in the ER. Next, we have the Golgi apparatus, which tends to be a lot of mystery for a lot of people that are learning about cells. Sure, they might know it exists, but they're not really entirely sure about what the Golgi apparatus does. It's really just where we're going to be modifying and transporting the products from the ER. So the ER is making a lot of protein and other molecules, but that's what it does. And so where does it need to go in the cell? Do we need to secrete it outside of the cell? Do we need to package it up and move it around to different parts of the cell? Is it used for structural stuff? And all that stuff will be kind of our packaging center, and our shipping center is our Golgi apparatus. So our manufacturing happens in the ER. Packaging and shipping is the Golgi apparatus, right? So they make little vesicles out of their membranes, that lipid bilayer again, little vesicles that are just little bubbles that will contain whatever the product is and then move it wherever it needs to go in the cell. And I think I've mentioned this before in this class, those vesicles aren't moving of a mind of their own. They're being carried along the cell's cytoskeleton by proteins. That literally these proteins will pop a vesicle on their back and walk it around like a backpack to other areas on the cell. So that requires a lot of energy. So always keep in mind, we're going to be making a lot of energy to just make things in the cell. We're going to need energy to run all of that. So Golgi apparatus, this is going to be our packing and shipping. Whereas our ER was going to be our manufacturing. All right, so this is basically going to create our assembly line. The nucleus contains all the blueprints to build everything. In the endoplasmic reticulum, we're going to be manufacturing from those blueprints. And then the Golgi apparatus is going to be shipping and packaging everything that got made there. Okay. So here's a picture of that nucleus where the genes are going to be coding for all of that. Those are our blueprints. All right, and then we're going to go into the ER. This is going to be for manufacturing, whatever gene we're looking at, whatever products. And then in the Golgi apparatus, this is going to be our packing and shipping. Does anybody have any questions about that process? All right, moving on. There's ways that we can make all of these products and sometimes even store them for later use. And these would be in our lysosomes and in our vacuoles. Our vacuoles can store things like water for later use or food or nutrients for later use and all sorts of things like that. Whereas our lysosomes typically contain molecules that we will use for fighting off bad guys. So it will contain all sorts of toxins and acids and digestive enzymes and things like that, that we can then merge those lysosomes with the vesicles that where we eat up and wrap ourselves around a bad guy, take it into our cell, called a phagolysosome, by merging that lysosome in with that phagosome. Zome means vesicle, okay? So we have the phagosome when we phagocytose a bacteria or a virus or foreign material from outside the cell, merge our lysosome with it, and release those toxic chemicals in there, creating that phagolysosome, which is a combination of the two, and then digesting down whatever it was that we ate into its parts. We can then use its parts for, our own cell purposes, or then excrete them from the cell as waste. Okay, and so that's how we would be dealing with things that we ingest, and is a big component of our white blood cells in response to infection. We'll learn about that more in chapters 15 and 16. All right, The mitochondria, this is the powerhouse, the energy generator of the cell. It has a lot of folds of membrane called cristae. And then it has its inside, what we would consider maybe its own sort of cytoplasm. And that inside area is called the matrix. As it turns out, the mitochondrial matrix has its own ribosomes. Very, very interesting. We're going to talk about what makes them interesting in a moment. Its own DNA and its own enzymes, okay? So it has all of its own components to make copies of itself within your cell. In fact, we might have genes that would tell your body that you need to make mitochondria, but all the genes to build new mitochondria are contained within the mitochondria, not within your genes. Very interesting. So what makes that so unique? These guys divide completely independently from the cell and can make copies of themselves as that individual cell would need. So it's, for example, you're taking up running, suddenly the cells in your muscles and your legs would realize that it needs more energy. So they'll make more mitochondria to compensate for that, right? And so your individual cells can build more mitochondria to make more energy. And these guys also have their own DNA separate from your DNA. And these exist in a circular chromosome shape. Now, what does that sound like to you guys? Bacteria, right? So they have their own genomes in a circular format. And they also have 70S, which is the size of ribosomes that they have, the same size as what bacteria have. I'm not going to ask you guys about 70 versus 80, but you should probably know that mitochondria have bacteria-sized ribosomes, whereas the rest of our cell has eukaryotic ribosomes, known as 80S. So these are signs that our mitochondria likely evolved from some bacteria or similar ancestor that we ate up a very long time ago, has maintained these aspects of that within them as it has continued to serve us as an organelle. So this is evidence of that endosymbiotic theory that we introduced at the beginning. Moving on, chloroplasts. Now these guys are really interesting. They're going to use energy from the sun to excite pigment molecules. And after exciting those pigment molecules, allow them to make a little bit of energy in the form of that molecule we learned about in chapter 2, ATP, adenosine triphosphate. So we'll get a little bit of energy, but just enough that now we can use that energy to move it back into making sugars. That's what I'm trying to think. My brain's not working in my favor right now. To making sugars like glucose. And so they're going to use a little bit of energy from the sun to make glucose. And then what are they going to do with that glucose? Well, it can be used to build parts of the cell, but really it's going to be shuttled back into this guy, the mitochondria. And so that's how plants are making their organic molecules, by fixing carbon into things like sugar, allowing for them to make organic molecules from carbon dioxide, which is an inorganic carbon source, from energy from the sun, and also allowing them to have glucose for them to create more, even a ton more ATP comparably by using that glucose for powering energy. So when we get into talking about aerobic respiration and photosynthesis in chapter 10, we're going to be talking about the fact that plants don't just have chloroplasts. They also have mitochondria that are going to be able to power a lot of those processes as well. So you're usually going to have these in tandem. So this is going to allow for photosynthesis in organisms that have organelles. We have bacteria that can perform photosynthesis, like cyanobacteria. In fact, probably the biggest contributors to the existence of oxygen in our atmosphere, way back when, before multicellular organisms existed. But they don't have chloroplasts. And we would know that because we know bacteria do not have organelles. Okay, so they would just do that photosynthesis on their cytoplasmic membrane. It's the only membrane they got, right? So for our eukaryotic cells, they can have all these little structures defined in there and designated to these functions. And for chloroplasts, photosynthesis happens here, all right? This is where we would be making oxygen as a product if we were plants or algae, for example. Ribosomes, we've already introduced the ribosomes. These are our little machines that are going to be reading that working copy of our gene, our messenger RNA, to spit out protein as a product. They're made up of ribosomal RNA. And depending on if you are eukaryote or if you are prokaryote, they have different sizes that we've already explained. That the 70S for the bacteria, the 80S for the eukaryotes. Yes.
And I just want to make sure I get this cemented in my head. They create proteins, correct?
The ribosomes?
Yeah.
Yes, so they're going to be building proteins out of amino acids. And we'll talk about exactly how that's going to work with our different types of RNA and everything when we get to chapter 6, but that's what they're for. So they're the machines that are going to be actively engaging in building proteins with amino acids. Good question. All right, so the cytoskeleton, this is going to be creating structure within the cell as well as a transport mechanism. So we can use our proteins to carry things along these guys. These guys also help orient the organelles within the cell. So we have some structure within the cell and everything's not just sloshing around and bumping everywhere. We have three major components of the cytoskeleton: the actin filaments, the intermediate filaments, and the microtubules. So these guys are basically like, these are the smallest and these are the largest. And I think you can kind of figure out where intermediate would fall in there. All right, so as far as the sizes, and these guys, again, going to be holding your organelles where they belong. They're going to be very engaged in your mitosis processes, and they're the highways that the rest of the cell is going to be using to move things around within the cell. All right, let's get into some of our groups of our eukaryotes. Here we have the kingdom fungi. Within the fungi, there's all millions of different kinds of species that can be grouped into either macro or microscopic. Okay, so our macro, these are the guys that you will be able to see with the naked eye and visibly identify them. Our microscopic guys, these guys can exist as unicellular organisms potentially, or they can group together into colonies, but these are our molds and our yeast. All right, there's different structures and terms we need to be aware of within our microscopic, what we call mycology, dealing with our fungi. The yeasts, then most of us can get a little confused. Yeast are not bacteria, they are fungi, all right? So eukaryotes, and they have a round to oval shape, and whenever they bud off of one another, they have a very distinctive look to them that we don't have very defined equal shapes like what we see with bacteria when they make copies of each other. With eukaryotes, when they're kind of budding off of each other, making their daughter cells, they make tiny little daughter cells that then grow after they are budding off. So that's our yeast cells, and I have a picture of that here in a moment. Our hyphae, this is a term referring to our long thread-like cells that we see often whenever we think of molds and how it looks like hairs kind of branching off of each other. Those branching things are called the hyphae. And then we have the mycelium. The mycelium is the intertwining mask of the hyphae that will make up the body or colony of the mold. So when we're looking at mold that like grows on bread or strawberries, that's very obvious that we can see the hyphae and the mycelium all at once. But when we're looking at things like mushrooms, they also have mycelium, where we're having all these branching and things going on, but it usually looks a lot like roots, like plant roots. But it's just the actual vegetative part of the fungi, whereas the mushrooms are actually just the reproductive structures of the fungi. And so that's where we get this idea of that mushroom colony that's up in Oregon that has these interconnected mycelium, root-like appendages that are all interconnected with each other over thousands of miles under the ground. It's the largest living organism on the entire planet because of that interconnectedness of the mycelium under the ground. Okay, so that's the mycelium. And again, would be differing depending on what type of a fungi you are. Some fungi can kind of switch between hyphae version and the yeast type morphology. Those guys that can switch between are called dimorphic. We're going to be talking about fungal spores. And these are things that like would be unicellular aspects of these fungi that are released into like the air and the environment for reproductive purposes. So here's an image of our yeast, and you would see like what the parent cell would be looking like, and then it might bud off a little daughter cell, again, that could then grow, and then this guy might start budding off before it's finished growing, and so on and so forth. This is called budding. It is very different from what we see with the bacteria, where we have binary fission, which we'll get into more detail on that in chapter 9. With fungi, they have different ways that they can receive their nutrients. We have the heterotrophs, so different eating, or they can break down different variety of substrates. They don't rely on just one. They don't rely on just vegetation, but they can break down a lot of variety of different things. We have the saprobes. These are the guys that break down dead things. And then we have the parasites. Parasites are organisms that live in conjunction with another living creature and cause harm to it. And so we know that this is the case with fungi because we know that ringworm is caused by fungi. We know that athlete's foot is caused by fungi and then it's causing damage to our own bodies as a result, right? So here's some images of some of what we've got going on with our molds. And if we look at them at a microscopic level and see what we call our fruiting bodies, our reproductive bodies that our mold is actually making, what's actually going on in that fuzziness that we see in our mold. For reproduction of mold, we can just simply break off a piece of the hyphae and put it onto a new piece of bread, and suddenly you can grow a new mold. Or that'd be fragmentation. Or we can have spore formation. So this can be sexual or asexual. I'm not going to get into the sexual ones, but we're going to be talking about a little bit of the details of the asexual. Sexual, it works pretty similar. Maybe not in practice as how we would do ours, but as far as like whenever their sexual spores are interacting with each other, we're talking about exchange of genetic information and creating new versions of the genetic results through that. But with our asexual reproduction, we can just break off basically an exact copy of ourselves, and that can float off through the environment to go create more mold. For our asexual spore formation, you can have two different kinds of fruiting bodies with our molds, okay? These are the sporangiospores and the conidiospores, which we also call conidia as a group. All right, so our sporangiospores easy way to kind of look at these guys, sporangiospores have a covering. And we have cleavage within inside of that covering, kind of breaking down into our single cells that will then burst open and release, right, from that covering. In the conidiospores, we have these little breakoffs that are happening without coverings. And this is really important for their structure to be able to just continuously kind of release off these spores from their fruiting bodies. All right, so when we're identifying these, if you have some sort of a fungal infection or we suspect there's a fungal infection, then what we can do is look at microscopic aspects as well as macroscopic aspects of these fungi in order to identify them. So what's the color when they're growing when we look at it macroscopically? Does it have hyphae? Does it have sporangiospores or conidiospores and things like that? So when we look at them microscopically, you could see covered aspects versus uncovered, and that these conidiospores could be very defining characteristics of what these organisms could look like, whether we have dandelion-shaped ones or ones that look like paintbrushes. Penicillium, for example, that we have used to create penicillin from over time, looks like a paintbrush, sort of whenever it's kind of like this, like with a big paintbrush sticking out, and all these little spores can break off of it. Whereas Aspergillus, another very, very common mold in our environment that can lead to opportunistic infections for people who have sinus infections, that Aspergillus can have, basically looks like a dandelion, but it's not covered, it's open, it's still a conidia, okay? So that would be able to have a different shape, and I could tell the difference looking at it microscopically that way. All right. Most human fungal infections occur through accidental contact. Most of the time, these fungi will just exist having their own purpose in nature. And now they have found a created niche inside of the human body where it's moist and warm and you're protecting it somehow. And the reason I bring up sinus infections, you can be allergic to pretty much anything, whether it's pet dander or just seasonal allergies from hay fever or whatever. And you get a lot of irritation and inflammation in your sinuses. Then mold from the environment can get trapped in those areas where we have that swelling going on, and it can grow in there. And if you're allergic to mold on top of that, and you have even more increased inflammation, you're just creating a nice happy house for that mold that's lodged itself in your sinuses. And now you can get balls of mold forming in your sinuses that may be very difficult to treat due to that inflammation and tissue that builds up around them. It may even need to be removed surgically. So this would be something where that mold normally doesn't belong there and it exists just fine in our environment, but has taken that opportunity of this situation to create a pathogenic presentation. So we call them opportunistic pathogens. Okay, so these are opportunistic infections. So we see these as being potentially community associated, where you could have spread of fungi from person to person, or hospital associated. Being in clinical settings can set your body up for being susceptible to certain kinds of fungal infections, bacterial and viral as well. But then we usually see these as opportunistic. This can be associated with certain situations like having a sinus infection or having reduced immune function like what we see with HIV. All right, here we have down here during the COVID pandemic, India struggled with something called mucormycosis. This is a fungal infection that killed more than 300,000 people with COVID. That having COVID lowered their body's ability to respond to the fungus, and they were therefore more susceptible to this. It's actually a serious, very serious infection, the mucormycosis associated with that. Moving on to the protists, this is essentially going to be that kingdom Protista. So Protista includes algae and protozoa. And a protist is any eukaryotic, unicellular, or colonial organism. So it could be multicellular, but we just don't have true tissues that are developing. So not true differential function of this, our liver tissue versus our, spinal tissue and things like that. We don't have true defined tissues here with these guys. So with the algae, probably most important guys are going to be the ones that are serving as algae within our marine systems. And this includes certain kinds of plankton that might be photosynthetic. These usually are the ones that are producing a good majority of Earth's oxygen since the beginning of time. So these are our algae and our first eukaryotes that were going to be our photosynthetic organisms. They're also important as far as creating things like red tide that can create toxic products that can affect our nervous system. Well, we won't really talk about them in microbiology. What we will talk a lot about are the protozoa. So protozoa, these guys are always single cell, and they're organelles, and they have specialized vesicles and appendages that work a lot like structures of multicellular organisms, even though they are single celled. So they might look like and behave like they have mouths, even digestive systems and reproductive tracts and legs, quote unquote, but they're really still just single cell organisms that have just utilized the structure of the cell for that functionality. These guys are protozoa. This is an R, I promise. Protozoa, these guys are heterotrophic. So remember what that means, different eating. So different food sources. So these guys can eat all different variety of different kinds of food sources. And we have free living versus parasitic. The free living guys will basically eat off of dead material or live off of bacteria or algae, whereas the parasitic guys live on other multicellular organisms and create harm as they're doing that. They may actively feed on tissues, for example. So we have different kinds of locomotion for these guys. We've already introduced the concept of flagella and cilia for locomotion of cells. So these are single-celled organisms that can move by using either flagella, by using cilia, or by using something called pseudopods. These are false feet, it's in the name. And these are what we think of with amoebas. We're just extending part of the cell out and latching onto a surface and then pulling the rest of the cell along that way. So using these fake feet, these outreaches of our cell to attach the surface and pull the rest of the cell along with it to maintain motility of that cell. So we are usually pretty aware of what an amoeba is and how the fact that it does goop along like that with its fake feet and its ability to move that way. We also have, so there's 4 groups. I want to be very clear, there's 4 groups of protozoa. The ones that use flagella, the ones that use cilia, the ones that use pseudopods. So these are our fake feet. And then the ones that are non-motile. And all of our non-motile ones are parasites. We have some flagellated ones that are, some pseudopod ones that are, and some ciliated ones that are, but all of the non-motile ones are parasites. Moving on, just like we had bacillus and clostridium that could produce those dormant structures, those dormant protective structures of endospores, we have eukaryotic protozoa that can do that too. And so our trophozoites are our motile, feeding, reproducing, dividing, metabolically active guys. So these guys are active. This is similar to the vegetative bacteria. Okay, whereas our cyst is similar to those endospores. These are very resistant. They are dormant. And this is going to be usually when you get infected with a protozoa, a lot of times you get infected with the cyst, and then it becomes the trophozoa, which is going to be producing the toxins and dividing and creating the illness within the person, right? Because when you're dormant and you're inactive and you're completely inert, you're not creating anything that would lead to illness. So always remember that about your endospores and your cysts. They're dormant, they're inactive, they're not causing the illness. The trophozoa or the vegetative forms will be. Okay. All right. Most protozoa, we can identify them by their appearance of the cell themselves, and we will characterize them based on size and shape, how they get around. Obviously, we can characterize them into those 4 groups just by how they are getting around. And then whether or not they make cysts or trophozoites and what those cysts and trophozoites look and behave like, and then where we find them in the body. So here are some examples of some important pathogens that are protozoa. So these guys are parasites. When we talk about parasitology, typically that term is referring to eukaryotic parasites. So our protozoa and our helminths, which we'll talk about in a moment, are worms. So these guys are our protozoa. For our amoeboid ones, these are the guys with the fake feet, the ones that move by pseudopods, right? So we have entamoeba histolytica that causes what we call amoebiasis. So this is going to be diarrhea and dysentery. Does anybody remember what dysentery was? We mentioned it before. Anybody know? So dysentery is bloody diarrhea. And this will come up over and over again. And hopefully not in your life, but in the lecture, we will have talking about this. Yeah, yes. So we want to avoid that wherever possible. But entamoeba histolytica can cause dysentery. It's very disturbing sort of presentation of a GI tract illness. And then we have these guys down here, which you guys are probably most familiar with as the brain-eating amoebas. This is going to be Naegleria fowleri and acanthamoeba. And these guys can actually be found in your water heaters and in your tap water. If you're not washing your hands and sanitizing your contact lenses well enough, you can actually get infections from these guys from your own tap water. So please be aware of that. But these guys, infection, brain-eating amoeba infection, extremely rare, extremely rare. We don't have very many cases of them from year to year. But if you do get this, it's pretty much a death sentence. We have a 97% mortality rate with these guys. It's very quick to happen. So you don't want to get the brain-eating amoeba, but it is very, very rare. Okay, so be aware of that. Balantidium coli is an example of our ciliated. These are those little oar-like appendages. So this guy causes GI symptoms. Then we have in our flagellated protozoa with our proper flagellar movement here, we have Giardia. You may have heard of this one. It's traveler's diarrhea. We have basically prolonged couple of months long, greasy malodorous stools. And this can actually become part of your normal flora for some people. If you've been exposed to this and recover from the disease, you can still maintain it as part of your flora. Trichomonas vaginalis is a flagellated STI. Then we have trypanosoma. We have Brucei and Cruzi. These guys are transmitted by biting insects. And so it's will be sleeping sickness, which was found in Africa, and Chagas disease, which is something we do have here in the United States infrequently, but it's transmitted by the kissing bug. We'll talk more about those diseases in later chapters. Then we have leishmania. Leishmaniasis is something that's usually transmitted by a biting fly. And that the biting flies, we saw a lot of outcome of this from our troops that were going to the Middle East, getting skin infections from the bites of these flies, transmitting this flagellated protozoan. Leishmaniasis. And then we have our non-modal protozoa. And again, all of the non-modal ones that exist are going to be parasitic in some form. So here we have the Plasmodium species. In particular, Plasmodium falciparum, these are associated with malaria, which is a blood infection. And these guys will kind of hang out in your liver cells and then cycle in between your blood cells. And every time they burst out, we have these massive expression of symptoms. We don't see a whole lot of this in America. Usually when we do see it in America, it's usually from people who have traveled to areas that have this endemically. But we do have the mosquitoes here in America that could potentially transfer this as a risk. So as we have increasing temperatures from global warming, it is something that we want to be sure that we're not having become endemic here. That means part of our normal disease presentation here as the US. Toxoplasmosis. So Toxoplasma gondii is something that's transmitted. Typically, it's carried by cats, so it's feline reservoir. And this is why we tell pregnant women not to handle cat litter because of toxoplasmosis. We'll talk about the effects of that and the symptoms and everything, again, in later chapters. And cryptosporidium, this is the diarrhea that you get from swimming in public swimming pools. So these cysts are very resistant to chlorine. And so with the cysts, you ingest them into your body from swimming in poopy water, which you might not know is poopy. But hey, if you've been, anybody in here ever been a lifeguard before? Yeah, so if you've been, you know then how much people are pooping in that water. It's probably a lot more than the rest of us realize. So these cysts, yeah, it's a lot more than you probably realize.
About one person poops in a thing like an hour.
Yeah, it's a lot. It's a lot. So
Bad.
it's usually, I mean, we say have loose stools. We all have it happen, but maybe don't swim in the public swimming if you have this issue or if your children have this issue. But the cysts can get swallowed and then they become trophozoites and cause disease in your own GI system and then gets spread through water and food that way. And then we have cyclospora, again, GI symptoms associated with that one. And there's others too. Moving on to our multicellular organisms. Okay, so these guys, the adults, are what we would call macroscopic. We can see the adults with the naked eye. But a lot of times when we're talking about helminths, we're talking about, these are our parasitic worms. These are multicellular and they are classified as animals, so kingdom Animalia. And that these worms, the adults may be able to be seen by the naked eye, but they're usually housed somewhere in the body where you're not going to see them. So how would we diagnose this? Well, we have symptoms that are associated with this, maybe signs of your white blood cell count that would indicate that you have these. And then we would look at your stool or your blood or tissue samples and look for the larvae and the eggs from these guys, that those are microscopic, and that's why they fall into microbiology, okay? So we can basically classify these guys based on their morphology. So that's like looking at them and how like their cells are arranged in their bodies. So we have our flatworms. So they have a flatworm presentation that we would divide into the cestodes, which are our tapeworms. And then our trematodes, which are our flukes. And we'll get into more info on these guys whenever we get into our disease chapters in Unit 4, when we talk about the diseases they cause. But this guy here, this is a fluke. And obviously, these here are examples of tapeworms or cestodes. Right? And then we have our roundworms, and these roundworms are also called nematodes. Flatworms also get a special name that isn't shown on here, but it is a name that you have to be aware of. Platyhelminthes. And I'll never make you guys write it, okay? But you do need to be able to recognize it. That if I ask you what is the scientific term for grouping our flatworms, that you would have to know that it is Platyhelminthes. And I remember it because plat sounds like flat to me. Right, so that's how I remember that. And remember, helminth is always going to be our multicellular parasitic worm. So the flat multicellular parasitic worms. The roundworms are called nematodes. All right, and they're unsegmented. So when we look at these guys, again, usually they are multicellular. They have multicellular aspects to them, including reproductive tracts and organs and mouth parts and things like this. So they are true multicellular organisms and they live life that way. They have very complicated life cycles that usually depend on multiple hosts in order to go through their life cycle. The host where they actually go through adulthood and have mating occur is called the definitive host. So that's a term that you need to be aware of. So anything in between is called an intermediate host. And these guys can have a lot of steps needed in order to get to where they can actually have mating occur. And we'll talk about those and how that works when we get to chapter 23. All right, so we can get these guys through food, soil, and water contamination, or by eating tissue from other animals. This is usually going to be through oral intake. Some of these guys can actually migrate into your body through your intact skin, like literally burrowing through your skin, like through the soles of your feet, or if you're swimming in water, burrowing in through the skin in the water. These are a lot more common than we also think. Here in America, we do have a few helminth infections, things like pinworms. Occasionally we'll see tapeworm infections, but a lot of these other helminth infections are incredibly common in undeveloped countries or underdeveloped countries, in areas where they don't have advanced healthcare, that we actually see quite a lot of helminth infections. Insects can also transmit these guys as well. And we'll usually characterize these based on how the larvae look in the microscope, the eggs, whether we're finding them in feces versus blood or urine, or arrangement of their organs inside of the adult, and all of those macroscopic versus microscopic differences, helping us break them up into if they are helminths, whether they are platy, the flatworms, or the nematodes, the roundworms. And if they're platy, whether they are cestodes like tapeworms or trematodes like the flukes. And there are just some examples here. Again, we're going to talk about some of these guys in chapter 23 when we get into the human diseases caused by these. So finally, we made it to your favorite part, the class questions, and we have time to go over them today. So get ready. Our first question, this is going to be for Jordan. What is the biggest difference between eukaryotic cells and bacterial cells?
So I'm not like 100% sure, but from what I have gathered from today, eukaryotic are multicellular and have more to them, while a bacterial has a nucleus, but it doesn't have as many like organelles as eukaryotic.
So close. Good line of thinking. Yes. Did you have a question? Okay, sorry. You're okay. I saw your hand. You're good. You're good. So eukaryotic cells have organelles and a nucleus. So the name actually means true nucleus.
Yeah.
So these guys, eukaryotes, have organelles.
Membrane bound.
And they have, exactly, membrane bound. And they have a nucleus, a true nucleus that is bound by a membrane. Bacterial cells do not have membrane bound organelles. They do not have a membrane bound nucleus, okay? So they do the same processes, but this is all happening without the organelles. It's just happening in the cytoplasm freely or along their cytoplasmic membrane, okay? So the biggest difference between these guys, eukaryotes have membrane-bound organelles, including a membrane-bound nucleus. Bacterial don't. Okay, so that's a big, big difference. And that's the one I guess I want you guys to lean on when we're talking about the differences between these guys and how we're grouping. And yes, eukaryotes can be multicellular, but they aren't always. Like our protozoa are unicellular. Okay, so that's not always the defining factor either.
I was just noticing you're very.
No, yeah, you're fine. That's a great line of thinking to get us thinking about that. And that's why we have these questions to help us think on that, because if you're thinking that way, other people are too. So our second question, this one is mine. What is the eukaryotic nucleus used for? This is the control center of the cell where we're actually housing our genetic material, our DNA. Our third question for Jessica, what is the function of the mitochondria? Jessica.
Energy.
Exactly. So that's going to be our energy maker, our powerhouse of the cell. Great job. All right. Next question, we have Reagan. Name the two subkingdoms in Protista. Great. So the algae and the protozoa. You guys didn't hear her answer. She got that right. And then our last question, this one's for Katie. What is the scientific term for flatworms? Platyhelminthes, exactly right. So that's it for lecture today. We have a little break until we get to lab. So I'm gonna go ahead and play the relevancy video. Remember, we have lab at 11:30. And if you guys want to wait here and watch the relevancy video, you're welcome to. But if you want to go take your break, you are welcome to do that as well. Yeah, you're fine.
What chapters tomorrow? I think I bumped up by.
War has
Oh, wow.
been going on for billions of years that breeds well-armed monsters
Sorry,
who struggle with other monsters for survival. Having no
There we
particular
go.
interest in us, most of them are relatively harmless, as our immune systems deal with their weapons easily. But there are exceptions.
Okay.
Negleria fowleri is an amoeba that has not only developed a deadly taste for human brains, but is also a match for our defenses and stars in dramatic headlines. What happens when this monster enters your body? Naegleria fowleri is an amoeba, a microbe with a nucleus, one of the smallest life forms on Earth.
Yeah.
It is a voracious hunter
That's
of
the problem with a lot of
bacteria and
people, right?
other critters that it devours whole and rips into pieces.
Science was my
Like
worst
many
subject.
amoebae, it is able to transform into different stages that help it survive. But most of the
But now
time,
I'm a nurse.
Naegleria fowleri is in its trophozoite stage, during which it looks like a squishy blob with tiny arms, and hunts, divides, and thrives. Its natural home is in freshwater, ponds, rivers, lakes, and hot springs. But unfortunately, it also feels happy in pipes, swimming pools, fountains, or spas when they're not properly treated. The warmer the water, the more it thrives and multiplies. So in the summer, when humans seek to cool off and enjoy themselves, the chances are highest that both species will interact. Because this makes it hard to avoid, millions of people regularly have contact with the amoeba, especially in warmer climates, and many people even seem to have antibodies against it. And this is mostly okay. You can even swallow it without consequences. Things turn bad when people dive or swim in water contaminated with the amoeba and water splashes high up into their noses. In a single drop of lake water, there are millions of viruses, bacteria, and amoebae, and that isn't really a big deal. But Naegleria fowleri is different. Let's zoom into the nose of an unsuspecting victim enjoying a great summer day and see what happens. First of all, the amoeba doesn't really want to be inside your nose, as it's not really looking for trouble. It just wants to eat a few bacteria. Instead, it's greeted by your natural defenses. Unfortunately for humans, Naegleria fowleri happens to be exceptionally good at generally flying under the radar of your immune system. For example, the inside of your nose is covered by mucosa, a slime filled with chemicals that kill or stun possible invaders or alert immune cells. But Naegleria fowleri is not particularly bothered by them, and instead calmly checks out the scenery, mildly annoyed about the whole ordeal. Now, if you are unlucky, the tiny critter stumbles over something that actually sparks its interest: nerve cells. Your nose is filled with a large network of olfactory nerve cells that pick up molecules from the outside and transmit their information to your olfactory bulb, the center of smell in your brain. To do their job, these cells talk to each other by releasing various messenger chemicals and recognizing them via specific receptors. One of the most important of these chemicals is acetylcholine. Through sheer evolutionary bad luck, Naegleria fowleri happens to have receptors that recognize acetylcholine, and it seems to attract them irresistibly, a little like moths that are attracted by night. So as your olfactory nerve cells do their job, using plenty of acetylcholine to talk to the brain, Naegleria fowleri enters your tissue. It seems to follow the chemical signals upstream. Neutrophils, crazy suicide warriors, begin to attack the amoebae. Individually, they have no chance against them, as the invaders are large and pretty buff fighters, used to dealing with tough enemies. So the defenders swarm the intruders and kill them either by vomiting chemicals that punch holes into them, or by literally ripping parts of them off and devouring them. But the Naegleria fowleri train is still on track, and while the neutrophil attacks slow them down, they continue to follow the olfactory nerves to their final destination, your brain. This process can take between 1 and 9 days, and you'll probably not notice anything during that time, until the amoebae reach the olfactory bulb, the center of smell and entrance to your brain. Your brain cells are nothing more than helpless victims, and they all release that wonderful acetylcholine. Naegleria fowleri initiates a massacre and releases an onslaught of various attack molecules. Some of them are basically little bombs that rip holes into your cells on contact so their pieces can be easily consumed. But Naegleria fowleri is now multiplying, and it's also becoming really creepy. In a feeding frenzy, it can develop up to a dozen suckers called food cups that look like giant, eerie mouths. The amoebae engage your brain cells, suck them in, and rip large bites out of them while they're still alive. Now things escalate quickly, and the disease that will kill you sets in. Alerted by the massacre, millions of immune cells, neutrophils, eosinophils, and microglias invade the infected tissue. Which is a problem. Your immune system is dangerous and not exactly a careful fighter. It's like burning down a forest to kill the wolves inside it. A really bad idea, I'm afraid. They waste no time and attack the amoeba, using all the weapons available to them, from chemicals to trying to eat them alive. Neutrophils explode themselves to erect barriers spiked with deadly chemicals. A fierce battle ensues. Naegleria fowleri can actually fight back, itself attacking and killing many immune cells. The immune system now throws everything it has at the invader, but in vain. The complement system, tiny protein bombs that can kill intruders on their own, are easily disabled. Antibodies, usually one of your superweapons, are just destroyed or swallowed. A high fever that usually slows enemies down does nothing, as the amoeba actually thrives in the heat. All the while, the amoebae continue to multiply, fight, and devour your brain cells. A disastrous chain reaction is taking place. One major thing your immune cells do when they fight is to cause inflammation, which directs large amounts of fluid from your bloodstream into the site of an infection. So as the battle rages on without a clear winner, more and more fluid enters the brain. At this point, the human will feel symptoms that quickly escalate. It all begins pretty vaguely: a headache, fever, nausea, and vomiting. As the battle spreads rapidly through the brain, serious symptoms appear, from confusion, inability to concentrate, to fatigue, seizures, and hallucinations. The brain swells up massively, but can't expand due to the bones surrounding it. So it compresses and disables the brainstem that controls things like breathing. Usually within a week, the patient dies. Up to 97% of the patients infected by the amoeba share this fate. In almost all cases, by the time an infection by Naegleria fowleri is recognized, the disastrous battle for the brain is already so far along that there is almost nothing to be done. Not only do we currently not have effective treatments, there are also an abundance of open questions about how an amoeba that usually enjoys its life in open water is able to overcome our immune system so effectively. So how worried do you need to be about this horrifying killer amoeba? Well, not very. While the Naegleria fowleri is clearly extremely deadly, and the infection truly horrible, there have only been a few hundred cases in the last few decades. You are way more likely to drown in a pool than to get infected. Not only does the amoeba need to be flushed high up your nose, it also needs to get a good grip, and it also has to make its way through the first lines of your defenses. Ultimately, Naegleria fowleri is neither evil nor a huge public health risk. But every year, some unlucky people have to deal with it. We still have so much to learn about it, and until we find a way to treat it, Naegleria fowleri will continue to be this vague and horrifying thing, hunting in puddles and lakes, and sometimes pools. Usually for bacteria, and very occasionally for people. Oh, hey, who are you?
It's existential threat.
Okay.
I'm here to make you question your own existence and everything you hold in your life.
Could you not, please?
Too late. Just look at all these amazing posters full of humbling.
Anyway, so this website, like this group on YouTube, they have a lot of really good science videos. I highly recommend you guys check them out. This is Kyrgyzstan. I just Kyrgyzstan. Anyway, look it up. They have really great videos. We'll show a few of them when we have time here. So we'll go ahead and head up to the lab now and get those randomly swabbed plate things that you guys grew out and look at those.
Okay, how are you? Week 3.
Yeah.
Hey, we got